Hello and welcome back, and this is lecture number 8, and I'm calling it Eukaryotic Ins and Outs. It is going to be about the plasma membrane of the cell and how materials get in and out of a cell. And I've got some funnies here that kind of remind you about the structure of that plasma membrane. It's a phospholipid bilayer, and over here on the left we see it's a phospholipid bilayer with the phosphate group at one end and the fatty acid tails coming off of here, which makes a hydrophobic interior region which doesn't like to let water through. So we're going to talk about how water, in fact, look, these are some like little water molecules right here, looking at it. This is about how different materials get through the membrane from one side to the other, which really controls a lot of the cellular functions. So these funnies are just kind of making jokes about that process. One of the things we're going to learn about is called passive transport, which lets things through the membrane, but in a controlled way. So this membrane is being what I would call passive aggressive. So it's like saying, so you want to pass through? Oh, sure, go right ahead. That's just fine with me. So it's kind of doing it begrudgingly. That shows that there's some level of control, and that level of control we use to describe what's called a semi-permeable membrane. It's letting some things in and not letting other things in, okay? What else do I have here? I've got a couple of, these look like minerals on opposite sides of a membrane, and it's a joke on Romeo and Juliet. Juliet is on the left saying, where are you? And Romeo is saying, I'm stuck behind this semi-permeable membrane. And then the last one, it's using the word semi-permeable again, letting some things through. And here's a thing that looks kind of like Romeo or Juliet. It says it's a salt. And there is this purple blob in the middle of the membrane. It's called a transmembrane protein. And you can see it kind of has a pathway through it. And it's saying, wait a minute, do you have an appointment? It's only letting some things through. And that's the semi-permeable membrane. Anyway, sometimes when you tell a science joke, you kind of got to explain it and all. And so that's what I was doing. So let's get started. Okay, so our first stop is with some terminology here that's used to describe the plasma membrane. We say that it is a fluid mosaic. Fluid because it's moving, and the things that are in it are moving around. Mosaic because it has lots of different parts that are like the tiles or the little pieces that a mosaic picture is made out of. So what do we have here in this picture? First of all, we have to recognize that this is the outside of the membrane and that this is the inside of the membrane, inside of the cell. And the reason that I know that, sorry about that, oh, that's pretty bad. The reason I know that is because inside we have filaments of the cytoskeleton. So that's the inside of the membrane. And we have some proteins that are embedded in the membrane. Again, you can recognize the phospholipid bilayer. But we have some proteins that are embedded in it. Some of them are on one side of the membrane, and some of them go all the way through. And I mentioned that those ones that go all the way through are called transmembrane proteins, as opposed to these ones on one side right here. That's called a peripheral membrane protein. So some of them are on the outside, and some of them are on the inside. And together we call this the fluid mosaic model of membrane structure. So what do we have here? Wait a minute. We have hydrophobic interior, hydrophilic exterior. We learned about that before. And then transmembrane proteins that are free to move about within that structure. Again, all of this is about maintaining equilibrium or flow from one side of the membrane to the other. All right, so we've got zooming in a little bit more detail here. We're really only talking about two things so far. We've got this phospholipid bilayer, which has the protein heads and the fatty acids, and has that hydrophobic middle, so we're not letting anything through, especially things that are dissolved in water. And then we've got this transmembrane protein. And if we remember how proteins are created, they are strings of amino acids, remember, from monomers of amino acids to polymers of polypeptides that then are folded up into proteins, into these complicated shapes that have certain functions. And in this case, this transmembrane protein it seems to have a little opening in the middle here that is letting things through. So that's one of the major functions of these transmembrane proteins. But there are other functions, and I just wanted to quickly mention some of the other functions, some of which we've already talked about a little bit. So the first one is transport, and that's what I was just mentioning. There can be transport that is just letting things through the middle of the protein. We call that passive transport because it doesn't involve any work. And then we have transport, which is kind of pumping things from one side to the other. And that requires some energy, some fuel. And what is the fuel inside of a cell? It is ATP. So this cell is going to be using some fuel in order to pump things from one side of the membrane to the other. The next function of transmembrane proteins is enzymatic activity. Sometimes there are proteins that start or stop a metabolic reaction. In this case, these enzymes are next to each other because they're creating a chain reaction of things that are happening. This little red triangle is coming in, and then it's causing this little blue square to be released, which is coming in here, and then it's causing this little green circle to be released. So just think of it as a chain reaction of chemical reaction controlled by these proteins in the membrane. Then we have a signal transduction function where some chemical is bumping into the protein on the outside of the cell, and it is causing a reaction that starts a series of activities inside the cell. And the activities usually involve the DNA and making new proteins. And so it's taking some information about what's going on outside and saying, oh, we better do something about this. And so we call that signal transduction. The next one is cell-to-cell recognition. Two cells that belong in the same body can recognize each other because of glycoproteins on the outside clinging to these transmembrane proteins. And you might have heard this with regard to viruses, because you've seen these pictures of like the coronaviruses with all these little things, spikes on the outside. Those are glycoproteins, and those are there to camouflage the virus, to actually have it bump into cells, and the cells do not recognize it as a foreign body. It's kind of like a lock and a key kind of thing, and that's how these cells function with cell-to-cell recognition, and then the viruses kind of use it in order to hide themselves. The next is something we've talked about, intercellular joining in a tight junction or a gap junction or a desmosome where the cells are clinging to each other. Proteins are involved in that. And then the last one, it says attachment to cytoskeleton and extracellular matrix. Lots of cells in the body are attached to other tissues. They're attached to the walls of blood vessels or the skin or the muscle fibers. And you see that attachment usually happens with association with these transmembrane proteins. And so we've discussed some of those things as well. Anyway, 6 different types of functions, but a nice picture of the transmembrane protein. All right, we're then zooming in, focusing in on one of the functions of these transmembrane proteins, and that is transport from one side of the membrane to the other. Again, I've mentioned all of these things. It involves the phospholipid bilayer, which regulates that passage. The hydrophobic, hang on, let me use my highlighter here. The hydrophobic layer stops water and the things that are dissolved in water, and the transmembrane proteins offer controlled doorways. One of the things you might notice with all these slides is that all the transmembrane proteins are purple, so that's an easy way to recognize them. Even in that first cartoon that I showed, there was the purple transmembrane protein that was saying, do you have an appointment? So I don't know why, but they've decided to make them all purple. And then, of course, the glycoproteins on the outside of another cell can establish identity so that we're taking things in or out of the correct cells and not just any old cell. The result is selective permeability. We're letting some things through and preventing other things. And we can do it in a way that just opens the door and lets them through that door, which is called passive transport. Or something which, if you open the door, they might flow in one direction, but we want to force them in the other direction. So we're going to use energy to pump materials from one side of the membrane to the other. And we call that active transport because it uses energy. It requires ATP. This picture here is, it looks to me like somebody made like a Valentine that involved cellular transport, which is kind of a weird concept, but look, it's all these hearts, and we have a cell, and basically it says, my selectively permeable membrane has let you through, so you've gotten past my filters, my defense mechanisms. I think that's what it means. Okay, so what we're going to do here is we're going to focus in on one of the types of transport. We're starting with the simplest type, and that is passive transport. Something is just being allowed to pass right through the membrane. I don't know what this something is. It is looks like a small blue hexagons, whatever kind of chemical that is. And there's a bunch of them on one side of the membrane and none of them on the other side. And we say that when things are transported passively, they follow a concentration gradient. That means that they go from high concentration to low concentration. They move in this direction. And so we're showing with this series of pictures here that we're getting more and more down here in the low concentration until they're balanced out. Now, I need to correct something on here because there's a pet peeve of mine that they say that they're moving from high to low abundance, but they're not just moving from high to low abundance. They are moving randomly, which means, there's the random right there. As soon as we have some over here, like these here, that means they are free to move randomly also. So there is lots of random movement, and so many of these may be moving in this direction, but these are also moving back. It's just that there's more on one side, so random movement results in a balancing out. It's not just things moving from one side to the other. It's random movement, so I like to point that out. When we talk about things moving randomly in a fluid, we call it diffusion, okay? And because we're referring to this as passive, remember that it cannot require any ATP, it requires no energy. So whatever these blue hexagons are, they are free to move back and forth. They probably aren't dissolved in water or attached to water because it wouldn't like letting water through that part of the membrane. So this is another representation of the same phenomenon. Remember, this passive transport, if it's random movement, we call it diffusion. And this is referring to diffusion of one solute and diffusion of two solutes. What we're looking at here is we got all these orange circles over here, and they're moving randomly, and because there's more of them on one side, they end up reaching equilibrium so that they're balanced out. In physics class, we would refer to this as entropy. Things that are organized tend to become less organized just simply by randomness in the universe. So if we have a situation where we have all of our orange circles on one side and all of our purple circles on the other side, they will both move randomly. This is showing that it's not about moving in one direction. They're both moving randomly and balancing out and reaching a state of equilibrium, balance. In order for that to happen, then both of these have to move away from a concentration gradient to balance out all of that movement. Diffusion. Oh, ha! I made a thing where the word diffusion was moving randomly, spreading out. That's good entropy. Good entropy. Okay, let's go on. Okay, well, that was the first type of passive transport right through the membrane, just going to let things right through. This is another type of passive transport. Passive because it requires no energy, no ATP, but we refer to it as facilitated transport because it's not just going right through the membrane, it's going through a special protein that's embedded in the membrane. This protein does not require any ATP, but if the protein wasn't there, it would not be letting things through. So we've seen a couple of these already. This is just another representation of that. We've got hexagons again, but this time they're green hexagons. So the first type of facilitated passive transport is a channel protein that just has an opening through it so things just flow through. When they're moving randomly, they go through this channel in the middle of the protein and balance themselves out because they're moving along a concentration gradient. The other type of facilitated transport, passive transport we have is something that is done by a protein called a carrier protein. And a carrier protein can only be open at one side at a time. So it's closed down here, but open up here. And then when something enters it, it will close here and open here. And so it's kind of, the door is letting things through, but it's not just letting them flow through. It's changing its shape in order to do this, but that does not require ATP. So this is still considered passive transport. So facilitated transport is a special kind of passive transport. And now instead of just the green hexagons, we've got some little green ovals or something that are entering in here and being moved through to the other side. Okay, so this is a slide that deals with a very specialized type of facilitated passive transport, and that is called osmosis. Osmosis is the facilitated passive transport of water from one side of a membrane to the other. Facilitated because it goes through channel proteins, transmembrane proteins. Passive because it doesn't require any ATP. And it's a very important process that controls where we find water, whether it's on the outside or the inside of a cell. Remember that passive transport is diffusion, so it's random movement of water molecules. But in this case, water molecules are polar. So they remember, they're sticky. They like to stick to some things. So sometimes they move randomly from one side of a membrane to the other, and then they stick to something that's there. And as a result of sticking to it, they can't get back across. And so it turns out to be random movement, but it's not back and forth. It's random and then sticking and then getting stuck. And that's what I've said here. A water molecule gets stuck to a solute and can't get back through the membrane. We say that that is osmotic pressure. We're getting water onto one side of a membrane and then it gets stuck there. So we have a picture down here. And in this case, whatever the solute is, that's these purple circles. Whatever is dissolved in water, we've got a few of them on one side and we've got a whole lot more on the other side. So what's happening is water is moving back and forth randomly. But as it moves back and forth randomly, when it gets to the high concentration side, it tends to stick to those and then not be able to make its way back. As a result, the pinkness is the water, by the way. As a result, we get more water on this side of the membrane because there's more stuff for it to stick to there. There's an example on the right here. These are actually blood cells. Red blood cells are kind of like squished hockey pucks. They look like a donut without a hole that goes all the way through. And those are normal red blood cells there. If we drop some red blood cells into water that doesn't have much dissolved in it, what happens is the water is moving back and forth across the membrane, but when it gets into the cell, it gets stuck. And so the cell starts to swell up with water and it builds up osmotic pressure inside the cell, and they're all blobby looking now. In fact, sometimes they may even burst because they get too much water inside. Just the opposite, if we take those cells and we put them in some salty water, water that is saltier on the outside than on the inside of the cell, what happens is the water is moving back and forth randomly, but it tends to get stuck on the outside and can't get back in. And as a result, those cells don't plump up, they shrivel. They're losing their water because the water is getting stuck on the outside. We do have some terminology associated with this here. An isotonic solution has the same amount of stuff dissolved in it inside the cell as outside the cell. So the water is moving back and forth randomly. There is no net osmosis. There is just water going randomly back and forth. The next type of solution is a hypotonic solution. Hypo means underneath. Hypothermia is getting too cold. You're getting under the temperature you should be. A hypodermic needle is a needle that goes under your skin. So hypo is under. Hypotonic means it has less stuff dissolved in it than the alternative. And so when we drop cells or organelles into a hypotonic solution, they tend to gain water. That's what's happening in this situation over here with the plump red blood cells. Wait a minute, I just have to put this in here. Okay, so they've been dropped into a hypotonic solution. Way less stuff dissolved in the water around them, so the water ends up getting stuck inside. Then the last word is a hypertonic solution. Hyper means greater than. Hyperactive is more active than usual. And so that means that there's a higher concentration of stuff in the fluid around the cells than there is inside. And so that's what we're looking at down here, where we drop these cells into saltier water, water that is saltier than the inside of the cells, and they tend to shrivel, they tend to lose water. I got one more picture here, and that is this guy who was in a movie you might be familiar with called Osmosis Jones. Actually, Chris Rock did the voice. This was an animated movie, and it is a great story, and it is a really fun ride through some biological processes. But let me just tell you, please, just enjoy it. It does not teach you anything about osmosis or about cellular function, okay? No science in it at all. Good movie though. Okay, well, so far we've been talking about transport, and we've talked about two types of transport. The normal passive transport directly across the membrane, and then we had facilitated passive transport that involves some transmembrane protein. And we've got both of those illustrated right here. Although I'm objecting to the arrows that only go in one direction, because passive transport involves random movement. So there ought to be at least a small arrow going in the other direction. So what we're going to do is we're going to add active transport to this. What makes active transport different? Remember that it requires energy, so we're going to have to use up some ATP, which is something we get from the mitochondria that it makes from the sugars. It is the fuel that we can use in order to do work in the cell. So the example that we have down below is the first type, which is what we call active transport. Remember, active is using energy. So primary active transport has got a transmembrane protein, but it's kind of a pump. It is grabbing things from one side and pushing them to the other side. And as a result, you can see that we're pushing them in a direction that wouldn't happen passively. We're building them up on one side. We're building them up where passive transport would cause that to balance itself out. So active means we're going to be going up a concentration gradient, not down a concentration gradient. We have a couple of other types of active transport here. I don't have pictures of them. Secondary active transport is when we move one thing from one side of the membrane to the other and we build up a supply of it. Sometimes we use that supply, which will flow back if we let it, in order to push something else along with it. That's why we call it secondary. We have to transport something to build it up on one side so that it takes other things with it when it flows back. So it says here, primary transport uses protein pumps, as I said, to move materials, and that secondary transport uses electrochemical gradients. So in other words, we've got a lot of this material built up over here, and as a result, it wants to flow back, and if we let it flow back, it can take some other things with it. All right, and then the last thing is bulk transport. I don't have any picture that illustrates that, but sometimes there are ways to just open up the membrane and let big chunks of things through. Let big things through that won't fit through a transmembrane protein, won't fit through the membrane at all, and we have a couple of terms associated with that. There's something called exocytosis, which is bulk transport out of the cell. Okay, exo is out. So exocytosis is out of the cellular, of the cytoplasm, exocytosis. And then endocytosis is into the cell. Exo is out, endo is in. And then we have something called phagocytosis, which is when endocytosis is happening to food, when a cell is eating, and there are some types of cells that eat, it engulfs something and takes it in. That's called cellular eating, and the word for that is phagocytosis. And then finally, there's another word here that is pinocytosis, which is which is basically, if we're going to call phagocytosis cellular eating, pinocytosis is going to be cellular drinking because it is endocytosis of liquids. So taking in solids is phagocytosis, taking in liquids is pinocytosis. And we'll see some examples of those. I just didn't have any pictures here. So remember, we did passive transport, and now we have 3 types of active transport. Okay, so this is one of those times where we have an example of something that is so common and so well studied that we say this is our primary example of a certain phenomenon. And the phenomenon is a combination of primary and secondary transport. So these brown things, I know I told you they were always purples. I guess I lied. So in this particular diagram, they're brown. But these brown things are transmembrane proteins. And what's happening is that on the outside of the cell, I know that because it says extracellular space, we have sodium ions, Na. We're familiar with sodium. And what's happening is that we have them outside because inside of the cell, we're gathering them up. Okay, we're gathering them up and we are forcing them to the outside. So that it seems like we have more on the outside. Remember, we're going up a concentration gradient. We're using energy because this is active transport, forcing them to the outside. What happens after we do that, see, we're using the ATP right here. We're showing using the ATP to do that. After we do that and the transmembrane protein is open, it's allowing a different kind of ion to enter in. This is the potassium ion. And then as a result of random movement, they come in there and then they are dumped to the inside of the cell. So sodium is pumped out and potassium flows back in. That is a combination of primary and secondary active transport. And it's just keeping these things on opposite sides of the membrane. Good example of active transport. Together we call this an ion pump because sodium and phosphate are both ions. And I find this so exciting, this ion pump, that I found another picture of it to show you the same exact thing. And this one we have the plasma membrane, the phospholipid, and in the big purple. We're back to purple transmembrane protons. Sodium is inside and it is entering the transmembrane protein and being pumped to the outside. And once it gets there, the potassium is flowing in and taking up those places and being pumped to the inside. So it's the same thing. I'm just showing it. It's stepwise and it's got a little bit more detail. Pretty cool. Ion pump. And this is my last slide and I'm at 28 minutes, so I'm doing it again. I'm getting in under half an hour. This last one is about bulk transport. So far we did 2 different types of passive transports, and then we did active primary transport, active secondary transport, and now active bulk transport. Active because this requires ATP energy, but it's not going through a transmembrane protein. What's happening is those little vesicles that are inside the cell are forming around material and they're carrying it to the membrane, or they're forming at the membrane and carrying things inside the cell. So we have our 3 different kinds of bulk transport here. The first type is called phagocytosis or solid endocytosis, and what we have is food particles, tiny little bits of organic stuff outside the cell are being gathered up and made into a vesicle and then transported, streaming along some little cytoskeletal fibers somewhere else into the cell, where then a mitochondrion will use them in order to make some ATP energy. So that is endocytosis, specifically phagocytosis, which is cellular eating. The next one is pinocytosis, which is the same exact thing, except it's liquids, not solid food, but liquids. And so we're taking in materials that are dissolved in water, and the vesicle is carrying water around instead of carrying food around. All right. The next one we're looking at here is endocytosis, that involves receptors, those little glycoprotein things that are on the transmembrane proteins. They gather up stuff. They recognize things and gather them up. And in this case, it's kind of like phagocytosis. It's taking in solid materials, but, by the way, this is receptor-mediated endocytosis. But you see that the materials are being grabbed by the receptor molecules, and then the membrane folds in and becomes a vesicle, and those things are trapped on the glycoproteins. So that's 3 different forms of endocytosis: phagocytosis, pinocytosis, and receptor-mediated. And then we have 1 form of exocytosis. That's getting rid of stuff, and it's just the reverse. All it is a vesicle which is moving towards the membrane, and when it meets it, a little opening occurs. And then the funny thing is that the membrane of the vesicle becomes the membrane of the cell. You can see that. So it just opens up, and this stuff is all released into the wild, into the area around the cell. So exocytosis. These are the examples of bulk transport. 3 examples of endocytosis, 1 picture of exocytosis, but they're all of them considered active bulk transport. I am done. See you in lecture number 9.